welcome back to What I Did in Lab Today, where I tell you guys what I did in my biochem lab today. So today was week 2 of our plant propagation lab. Last week we made a bunch of regeneration and rooting media. This week we actually planted our tissue samples. So for the rooting media, we had some samples of peppermint plants, and for the regeneration media, peppermint doesn't work great for regeneration, so we used some lavender plants instead. So first off, we had to actually collect our tissue. For the lavender, we just picked, I say we, but it was only me because my lab partner is not here this week. So we picked the leaves off of the lavender plant, and then that's all we need to do. For the peppermint, it was a little bit more complicated. Here, let me kind of show you how we would cut on one of my coleus plants. I'm not actually going to cut it because I like my plants, but I'll show you what we would kind of do. So here we have my floor coleus. Let's take this section for example. We want these sections, focus. These sections right here, we're not focusing, but this is called a node. This is where the leaf extends from. This is the part we want. So what we would do if we wanted to propagate this section, we would cut this section off and then trim so we get all of these nodes where the leaf comes out. So what we would do, cut here, and then cut this section of leaf off so you get just this node section. And then move up the stem, do it again on this one, do it again on this one, do it again all the way up, so you have a bunch of sections of stem with these little node bits coming out, but without the actual leaves. So, as we could see, our stir bar was behaving quite a lot better than last week. It was actually stirring properly. So, what's in those beakers? Sorry, not beakers, flasks. What's in those flasks is our plant tissue samples and a diluted solution of water and bleach, which works to kill any fungal or bacterial cells that have been on the plant tissue samples that we don't want. So what I had to do was leave the plant tissue in the solution long enough to kill off all of the contaminant cells, but without damaging too much of the plant tissue. We also had our negative controls, which are unsterilized samples of both the lavender and the peppermint. These ones have not soaked in bleach, they're just there to act as a control and to see kind of what contaminants we get from the just regular unsterilized plant tissue. After the plant tissue has cooked long enough in the bleach, we had to do a couple rinse cycles where we basically just get rid of the bleach solution, add a bunch of water, stir that around for a couple minutes, and then get rid of the water, rinse and repeat until most of, ideally all of the bleach solution is gone or diluted enough that it won't be a problem. Then we had to take out the plant tissue, cut a few areas off just because the bleach did kill a bunch of the tissue kind of on the ends of the samples. Because it is bleach, it is toxic. So we had to remove the dead and damaged tissue there. Then the samples were ready to put into the growth media, like you can see here in our negative controls. For the lavender leaves, they were put in the regeneration media. You just kind of stuck the tip of the leaf, the bottom tip, into the media and then kind of smushed it down flat with the tweezers in order to get the entire surface of the leaf lying flat on the media. And then for the rooting media up there into the little jars, just had to grab it with the tweezers and stick it right down into the media, kind of standing up straight. And since we spent all of this time sterilizing all the tissue, we really wanted to make sure that we don't get any external contaminants, so it was really important to maintain a sterile environment, so wear gloves, wipe everything down with rubbing alcohol, use a Bunsen burner to create that kind of sterile bubble. Make sure that you don't touch the pipettes and the tweezers and the scalpel to anything. If you do, you need to sterilize it again in the Bunsen burner, light it on fire, get all of those bacterial cells off. Make sure you keep your entire work area within that sterile bubble so you don't get any bacterial contaminants, or as small amount as possible, because again, this media is really good for growing things. But yeah, now these guys are going to incubate for a few weeks, and they ideally will grow into new peppermint and lavender samples, hopefully without any contaminants except in the negative controls, in which case there most likely will be contaminants, but I will probably report on the results in a couple weeks when they are done cooking.